This is a video for days 8 and 9 of the short read sequencing analysis workshop. This is video 4, counting reads to estimate gene abundance using feature counts. In this video, we're focusing on genes, which we are defining as the union of all associated exons. Generally, there are two main steps to estimating gene abundance. First is read counting, and second is normalizing those count values. Though this may seem straightforward, there are many aspects of transcription that can complicate this process, including 5' and 3' UTR boundaries, alternative splicing, etc. The first step requires that the reads have been confidently mapped to a location in the genome, and that the boundaries and labels of your features are well defined. We call this information the genome annotation. For count normalization, there is no one correct way. There are numerous methods, all of which have trade-offs. However, normalization is critical in order for you to draw any kind of meaningful conclusions from your analysis. The simplest counting procedure is just to count the number of reads that have some overlap with the region defined in your annotation. This process does not distinguish between a read that overlaps by only a single base and a read that lies completely within the annotated region. In the diagram you see here, the total counts for the genomic feature is the total number of green reads. The red reads, having no overlap with the genomic feature, are not associated. The next step, normalization, is essential because the number of reads that map to a feature depend on more than just the expression level of that feature. In particular, the number of counts assigned to a feature will depend on 1, the total number of reads sequenced, and 2, the length of that feature. The various normalization schema attempt to account for one or both of these factors. The simplest normalization method is referred to as reads per millions mapped, or RPM, and it's calculated by dividing the total counts for each feature by the total number of millions of mapped reads. This accounts for the differences in sequencing depths between libraries. A second method is called reads per kilobase per millions mapped, or RPKM, which will also account for the length of features in addition to the library size. To calculate it, the RPM values are subsequently divided by the length of the gene or feature in kilobases. RPKM can be used to compare between different genes within the same sample. Finally is transcripts per millions mapped, or TPM. This is an alternative to RPKM, in which the counts per feature are first divided by the length of the features, resulting in a reads per kilobase value. These RPK values are summed over the entire sample, and the resulting sum is used to scale the RPK value for each feature individually. What is convenient about TPM is that the sum of all TPM values within a sample is the same across samples, thus allowing for better comparison between samples. These are just a few of the most common normalization schema. Again, there's no one correct normalization method. The one you choose will depend on what comparisons you're making and what tools you choose to use for your analysis. One method for performing read counting over features is called feature counts. It is a part of the rsubread package for R, and as such works seamlessly with many of the common downstream DEA tools such as dseek2 and edgeR, which are also based in R. The inputs for feature counts are 1, a SAM or BAM file containing for each sample, containing mapped and sorted reads, and 2, an annotation file containing features and metafeatures, which is in either a GTF or SAF format. The output of feature counts is an R data frame integer count table with rows corresponding to your features and columns corresponding to your samples. Feature counts will perform read counting or summarization at feature or metafeature level. Feature counts defines a feature as a contiguous region of the genome, which in this case would be an exon. It defines a metafeature as an aggregate of more than one feature, which in this case would be a gene, or a union of multiple exons. The boundaries and labels for these features and metafeatures must be provided in the form of an annotation file. In feature counts, a read is said to overlap with a feature or meta-feature if there is at least one base shared between them. A read is multiple overlapping if it overlaps with more than one feature or meta-feature. Now let's discuss feature counts inputs in a bit more detail. Aside from the BAM files containing your reads, feature counts requires an annotation file. There are two common formats for the annotation file. One is the gene transfer format file, or GTF. It has 9 tab-delimited columns, including sequence name, commonly the chromosome ID, feature type, such as an exon, start codon, transcript, start position, stop position, strand, and then the 9th and optional column contains a lot of metadata that can include things like gene and transcript IDs. The other format is the simplified annotation format file, or SAF. As the name indicates, this format is a simplified version of the GTF and has a very similar format to a bed file. It has 5 required fields: gene ID, chromosome, start position, stop position, and strand. Feature counts has many options to tailor its read summary. Here's a list of some of the more relevant options. It's recommended that you read through the available options to know which are appropriate for your data. At minimum, you will need to specify an input vector file paths, an annotation file, whether or not the annotation file is in GTF format, whether or not to perform summary using meta features, i.e. gene names, and finally, whether or not your data is paired end. Now let's look at an example of how you might call feature counts. Feature counts is run in the R environment. So to begin, one would execute R on the command line or use something like RStudio. In this example, we have 10 BAM files to summarize. In the variable BAM file list, having already been specified, is a vector of strings, each of which is a total path to the corresponding BAM file. Once the rsubread library has been loaded, we specify some ID label for each sample. Here we've labeled them pH1 to pH10 and store these in the vector sample IDs. The vector is then converted to a factor to later be used for DEA. At this point, we call feature counts, assigning its output to a new variable called output. For our annotation file, you see we've specified the absolute path to an annotation file in the SAF format, and specified the value false for the GTF annotation file variable. Finally, we assign the sample IDs to the column names of the count tables contained in the feature counts output. Now let's look at the output we've created. The primary component of interest is the counts table. Looking at the contents of this counts table, we see it is a matrix of integer values, where the columns names are the sample IDs that we created on the previous slide, and the row names are the features or meta-features we indicated feature counts should summarize. In this case, the row names are gene IDs. Thus, the integer values are the numbers of reads assigned to a particular gene for a given sample. The counts matrix is the minimum necessary input for downstream DEA tools such as DE-seq or EdgeR. Here we've discussed one R-based method for calculating read counts. However, there are many others. For calculating counts over bed files, BedTools GenomeCov is available. For doing gene-wise counting similar to what we demonstrated here, there's the Python-based package htseq and another R-based routine called summarizing-featureCounts. If you're attempting to calculate abundances of transcripts, stringtie and sailfish are two popular options. It's recommended that you research the pros and cons of these options to determine which counting method will most be suitable for your data.